Good morning. Today's topic is gene transfer method in plants. Production of transgenic plants. Transgenic plants means genetically modified plants. They are actually done through the technique recombinant DNA technology. Through recombinant DNA technology, we can introduce foreign genes into the plants, and thereby the plants can act beyond its natural ability. The basic steps of RDT is discussed in the chapter 1. Now we will see the two ways by which the DNA engineering is done in plants. One method is vector mediated, where we use a vector. A commonly used is Agrobacterium, and Agrobacterium is also known as natural genetic engineer. And the other method is vectorless method. Here we are directly introducing the DNA into the plant cells without a special vector. In this picture it is shown particle gun. Particle gun is gene gun by which we are bombarding the genes onto the cells, and then the genes get integrated with the chromosome, and through plant tissue culture we generate plantlets. In today's class, we are going to discuss the first method in detail, that is Agrobacterium mediated gene transfer. Agrobacterium tumefaciens is a bacteria having a plasmid. Plasmid is Ti plasmid. This bacteria actually cause a disease, crown gall. It's a type of tumor. In plants, they generally forms tumor. If a plant is having cut or wound, bacteria infect here and create a tumor. Through many experiments, scientists revealed that this bacterium is having a special plasmid known as Ta plasmid. This plasmid is the vector, and that vector carries certain genes to this plant and make modification in their cells, and that leads to the formation of crown gall. That's why Agrobacterium is known as natural genetic engineer. Now we will see the structure of Ta plasmid. This Ta plasmid is having a region known as tDNA. Actually, at the time of infection, suppose this is plant cell and this is our Agrobacterium tumefaciens. At the time of infection, this Agrobacterium creates a copy of its tDNA, and that is actually sent to the plant cell. This gets integrated with plant's genetic material or chromosome and creates crown gall. So the portion tDNA is actually the DNA that gets transferred from Agrobacterium to the plant cell. Let us discuss the major steps in this process. The first step is disarming of vector. From the Agrobacterium, we isolate the Ta plasmid. We will insert our gene of interest into the Ta plasmid. This Ta plasmid is having a tDNA which is engineered. This tDNA have some virulent genes. These virulent genes are actually removed, and on that place we have kept our gene of interest. So this is the engineering of Ta plasmid, or we can see disarming of vector. Why disarming? Because they have some virulent gene that has been removed, and this is recombinant DNA plasmid now, to which we have added our gene of interest. We will add some markers like NPT2. This is a kanamycin resistant gene. Now the second step of this process is co-cultivation. That means we take out the tissues which are grown in plant cell culture, and the tissue and Agrobacterium which is containing recombinant Ta plasmid vector are grown together. We take the plant cells, and to which we will make some damages. Why we create some damages? So that the bacteria can easily infect. This is known as co-cultivation. Now the plants are transferred on shoot generation medium. In this medium, we will add the hormones for shoot generation as well as kanamycin. We add a kanamycin so that only those cells to which our gene is incorporated can only grow in the medium of kanamycin. We have incorporated a gene which is NPT2 that will have kanamycin resistant property. See, along with this, we add another bacteriostatic agent, that is cefotaxime. See, the agrobacterium is no more required now because already our gene has been reached the plant cells. Now, to kill agrobacterium, we add cefotaxime. To select the cells, we add kanamycin. For shoot generation, we add hormones. Now, the cells are kept in the shoot generation, then later on to the root generation, is rooting, then transplantation, that is hardening. Okay, before going to the transplantation, we can also confirm the presence of our gene of interest in these the cells by using certain methods. We can go for PCR, we can go for RAPD, or we can go for any other molecular techniques by which we can check whether the cells are having our gene of interest or not. These plants will be studied for their expression of our induced gene. A quick recap: at first we have selected Agrobacterium, we isolated TA plasmid. What we do, we done RDT, we isolated TA plasmid, and now we are doing recombinant DNA technology. Through recombinant DNA technology, we engineer the plasmid by incorporating markers like NPT2, remove virulent genes, we add our gene of interest. Plasmid is kept back to the agrobacterium. Through plant tissue culture, we make cells. In the co-cultivation, try to infect bacterium which is having TA plasmid, that is genetically modified. We will select the cells, and those cells are sent for shoot generation, then rooting, and plant regeneration. The confirmation of our gene of interest by using many screening methods. So altogether we are getting a transgenic plant. Thank you.